Hello everyone and welcome back to another A Level Biology video. So today's is on protein synthesis and this is for EDEXCEL A Level. So that's EDEXCEL AS and A Level Biology A, also called SNAB, and EDEXCEL AS and A Level Biology B. So our first specification point is for all of the EDEXCEL specifications. So this is to understand the formation of polypeptide and proteins, amino acid monomers linked by peptide bonds in condensation reactions. So here we have our amino acid monomers, and protein synthesis takes place, and then these amino acids are joined by peptide bonds to form a polymer, which is the protein. So next we've got some further biology A specification points. So number one is to understand the process of protein synthesis, transcription, including the role of RNA polymerase, translation, messenger RNA, transfer RNA, ribosomes, and the role of start and stop codons. And number two is to understand the roles of the DNA template, antisense strand in transcription, codons on messenger RNA, and anticodons on transfer RNA. And then the EDEXCEL AS and A Level Biology B specification points. Number one is to know that a gene is a sequence of bases on a DNA molecule coding for a sequence of amino acids in a polypeptide chain. Number two, know the structure of mRNA, including nucleotides, the sugar phosphate backbone and the role of hydrogen bonds. Number three, know the structure of tRNA, including nucleotides, the role of hydrogen bonds and the anticodon. Number four, understand the process of transcription in the nucleus and translation at the ribosome, including the role of sense and antisense DNA, mRNA, tRNA, and the ribosomes. Number five, understand the nature of the genetic code, including triplets coding for amino acids, start and stop codons. So for our first specification point for biology B, we've got our DNA molecule here, and we've got our gene selected. So remember that a gene is a section of DNA that codes for a specific protein. So a gene is made up of a sequence of bases, and this is the code. So the bases in DNA are T, A, C, and G, and they form a sequence, and this is what determines the sequence of amino acids, which then determines the polypeptide chain and the protein produced. So for our second specification point for biology B, we've got our mRNA strand here, and we need to know the structure of it. So mRNA has a sugar phosphate backbone which forms the nucleic acid structure composed of nucleotides. It has a backbone composed of alternating sugar and phosphate groups, and this defines the directionality of the molecule. So remember that nucleotides are made up of pentose sugar, a phosphate group, and a nitrogenous base, and this is what forms our nucleic acid. So the nucleotides are the monomers which come together to form the polymer of nucleic acid, which it can be DNA or RNA. So mRNA also has codons, which are also called triplets of bases, and these code for specific amino acids, and also hydrogen bonds form between the DNA bases and the mRNA bases during transcription. We've then got our tRNA structure. So it is a folded structure with three hairpin loops, so it almost looks like a clover leaf structure. And then it also has an amino acid binding site. This is where the amino acid binds, as you can see there. We've then got an anticodon, so this complementary base pairs with the codon on mRNA. Then we've got hydrogen bonds and our nucleotides. So tRNA is made up of nucleotides which are hydrogen bonded together in complementary base pairs. We've then got our points for biology A and biology B in describing the process of protein synthesis. So transcription is the first stage of protein synthesis, and this occurs in the nucleus. So here we've got our DNA molecule, and an enzyme which is called DNA helicase actually breaks the hydrogen bonds between the DNA strands, between the complementary DNA bases on these strands, and the DNA molecule unwinds. Now, within the specifications for all the A levels, it actually doesn't mention DNA helicase for protein synthesis, it only mentions it for DNA replication. So I would check with your teacher to see as whether you need to know about DNA helicase for protein synthesis, or only know it for DNA replication. Some of the specifications also put in RNA polymerase as this enzyme, even though that's not actually the one that does break the bonds, but they just use that as it's used further on in the protein synthesis process as well. So make sure to check with your teacher for that. So after the hydrogen bonds are broken and the DNA molecule unwinds, the DNA strands separate, and we've got our sense coding DNA strand and our antisense non-coding / template DNA strand. So it is in fact the template DNA strand, also known as the antisense or non-coding DNA strand, that is used for copying of the code to mRNA. So the antisense strand is almost like a reflection of the code from the coding strand, and then this is reflected onto the mRNA, and the actual code is therefore copied. So once we have our antisense strand, we've then got RNA polymerase lining up 3 complementary RNA nucleotides alongside the template strand, and hydrogen bonds form between the bases. So just a reminder that the DNA bases are A, T, C and G, and the RNA bases are U, A, G and C. So remember that the difference between DNA and RNA is that RNA has a U and DNA has a T. So in terms of the bonds that they form, DNA's A base hydrogen bonds with U, and that's for 2 hydrogen bonds, and DNA's T base hydrogen bonds with A, and that's also 2 hydrogen bonds, and then C hydrogen bonds with G, and then G hydrogen bonds with C, and those are both 3 hydrogen bonds. So once all the RNA nucleotides are aligned, hydrogen bonds between the bases break and RNA polymerase joins the mRNA nucleotides together, forming the pre-mRNA strand. Now, although it is a formation of a pre-mRNA strand, I know that definitely in Biology A they just prefer that you say an mRNA strand, and also Biology B doesn't mention splicing as such in this specific part of the specification. So again, just check with your teacher whether they think it's necessary for you to put pre-mRNA or mRNA at this point. So mRNA then leaves the nucleus and enters the cytoplasm for translation, and it attaches to a ribosome in the cytoplasm. So we need to know the role of ribosomes. So ribosomes are actually the site of protein synthesis. This is where translation takes place, and they attach mRNA, and then tRNA brings amino acids to form the polypeptide, and the ribosome catalyzes the formation of peptide bonds between the amino acids that tRNA brings. So here we have our ribosome, and our mRNA strand has attached, and then tRNA carrying the corresponding amino acid complementary base pairs to mRNA's start codon, and this is AUG, and it complementary base pairs with its anticodon, which is UAC. And then a second tRNA molecule carrying a corresponding amino acid complementary base pairs to mRNA's second codon with its anticodon. So these amino acids are actually coded for by the mRNA strand, as obviously the mRNA strand is a copy of the original DNA code in the gene. So tRNA continues to bring amino acids to the mRNA on the ribosome until it reaches a stop codon, and the polypeptide chain of the amino acids then detaches from the ribosome. So just moving on to our 5th point for Biology B. So here we've got our gene selected again once more, and as we said, genes are made up of bases A, T, G and C, and the genetic code actually includes triplets. And these are 3 base sequences which code for particular amino acids. So for example, we've got TAC here, and this codes for a specific amino acid. Then CGA, this is coding for another amino acid. TGC, coding for another one, and so on. So now we're going to move on to some questions based on that information. So we're going to start with Biology A SNAB questions. And number 1, leptin is a protein hormone with a role in the control of appetite in humans. The leptin gene is located on chromosome 17. State what is meant by the term gene. So we've got to describe what gene is here. So we know that a gene is actually a sequence of bases or nucleotides in DNA. So for the GCSE specifications, all you need to say is that a gene is a section of DNA that codes for a particular protein, but for the A-level specifications they want you to be a bit more specific. So you need to say that it's a sequence of bases or a sequence of nucleotides in DNA that actually codes for, you can say, the primary structure or the amino acid sequence or the polypeptide. They do allow that you saying that it codes for a protein, but it's better to be more specific just in case in further papers they don't allow you to say that. So gene is a sequence of bases in DNA that codes for the primary structure. And then we've got our second point here. So describe the role of tRNA in the production of leptin. So we've got the role of tRNA. So we know that tRNA is involved in translation. So that is obviously what they are asking us to discuss here. So we know that the tRNA actually carries the amino acid to the ribosome, and then it has an anticodon that binds to mRNA's codon. And then it carries a particular amino acid to the mRNA on the ribosome, which is coded for by the mRNA. So we can see our answers here. So question 2, a biotechnology company is developing a method of repairing the mutations in the gene for enzyme G. The method being developed is called CRISPR-Cas9. In this method, a short sequence of RNA binds to the DNA containing the mutation responsible for MPS I. This RNA acts as a guide to enable the Cas9 enzyme to bind to DNA. This enzyme can then be cut and repair the DNA, removing the mutation. Describe how scientists could produce the short sequence of RNA needed to treat someone with MPS I. So again, we've got a lot of information for this particular question, but all it's asking us is how we can produce RNA nucleotides at the end of the day. So here it says, describe how scientists produce a short sequence of RNA. So RNA is, of course, a nucleic acid. So in order to produce it, we need to transcribe the part of the DNA which contains the mutation, and this is going to produce the mRNA structure. And then we can use the RNA nucleotides to form this mRNA structure. And we can see our answer there. Question 3. A central idea in biology is that DNA codes for the synthesis of proteins from amino acids. The instructions for making proteins are in the form of mRNA. The diagram shows 2 stages in the production of mRNA. So although I haven't mentioned splicing in this particular presentation, this is actually included at a later point in the specification for Biology A, rather than at the specific point where the actual process of protein synthesis is discussed. However, a lot of the points for this question are just linked to transcription, which we've talked about already, so we can still complete this question. So from the diagram you can see we've got our gene, and then we've got our pre-mRNA. So the gene to the pre-mRNA is obviously transcription and translation, and then the pre-mRNA to the mRNA is the splicing which occurs. So this is the removal of introns. So within the pre-mRNA, we've got exons and we've got introns. So exons are coding regions and introns are non-coding regions. So splicing allows for introns, which are the non-coding regions, to be removed. So then our question is, explain how this molecule of mRNA is produced. So of course we know that we're going to start with transcription. So of course we know that the DNA unzips, and then one strand acts as a template, and then nucleotides pair up, RNA nucleotides pair up with the complementary bases on the gene, on the template strand, sorry. And then RNA polymerase joins these nucleotides together in order to form pre-mRNA. And then we can say that the mRNA actually has fewer bases than the pre-mRNA or the gene, because the exons and the introns are then removed and the exons remain. So as a result of the removal of these introns, mRNA is therefore only made up of exons. So even if we hadn't explained splicing to you, the diagram actually shows it very clearly that in fact these introns are removed, so you don't even really need to know what they mean. You just need to look at the diagram for this. But here we have our answer. Now this is from a sample assessment, and it does say ribonucleotides, but actually in the further papers you don't need to say ribonucleotides, you just need to say nucleotides or RNA nucleotides. Question 4. Describe the roles of transcription and translation in the synthesis of a globular protein by a muscle cell. So this question is basically asking us to describe protein synthesis, however we do need to link it to the specific protein. So just highlight the keywords here. So we know that protein synthesis begins with transcription. So transcription obviously begins with the gene or the sequence of DNA being transcribed. However, we need to say that in fact it's the gene for this protein that is transcribed. So that is where you need to be specific. We can then say that complementary base pairing between the RNA nucleotides and the DNA occurs in order to produce mRNA. And then once that's produced, transcription is complete, and then translation begins in the cytoplasm. So the mRNA leaves the nucleus and it attaches to a ribosome. Then pairing between codons on mRNA and anticodons on tRNA occurs, as we know, and the tRNA carries a specific amino acid coded by the mRNA to the mRNA on the ribosome. And the sequence of codons or bases determines the sequence of amino acids, which then determines the primary structure of the protein. And we can see our answer here. Question 5. DNA is a double-stranded molecule. During transcription, the antisense and sense strands are separated. Part of the antisense strand with base sequence TAC, GCT, GAC is transcribed. State where transcription occurs in an animal cell. So we know that, of course, transcription occurs in the nucleus, as this is where the gene code is transcribed. So, nucleus. Question 6. Which row shows the correct sequence for the complementary sense strand and the mRNA produced in transcription? We've then got some sense strand codes and then some mRNA codes as well. So we know that for DNA, the bases included are A, T, G and C, but for RNA or mRNA itself, the bases are A, U, G and C. So T is not a base of mRNA. So if we have a look at A and C, we can see that mRNA actually includes T bases. So we know that A and C mustn't be correct because mRNA does not have T bases. So we can just put a little cross through those. So then let's have a look at B. So remember that we're looking at the sense strand, which is the coding strand sequence, not the antisense template strand sequence. So if we write out the bases, so we've got TAC. So that means that the template strand that complementary base pairs with the sense strand is going to have the code A to T, and then A matches with T, and then C matches with G. So this would mean that the mRNA structure would begin with U, A, C. However, for B, we can see that we have got AUG, not UAC. So we know that this mustn't be correct. So B is evidently not our answer. So we can cross through that. Now let's just check that D is our correct answer. So we've begun with ATG, so we can write that down. So the template strand code is therefore going to be T, A, C, which means that mRNA should be T to A to U, and C to G. And we do have AUG here. And if we continue to go along the sequence and work out the template strands and then the mRNA code, we would find that these two do match up and they are correct. So therefore, D is our correct answer. And we can see our answer there. Question 7. A molecule of tRNA is made from a precursor molecule that is modified. Modification includes splicing, trimming and attachment of new nucleotides. The diagram shows a precursor molecule for a tRNA specific for the amino acid proline and a tRNA molecule specific for the amino acid proline. Some of the bases are shown in each diagram. So the first question is, which bond is labelled Z? So we know that tRNA molecules are made up of nucleotides, and these nucleotides are bonded together by hydrogen bonds. So therefore our answer to A is going to be hydrogen bonds. So we can highlight that there. And then next question, which of the following base sequences is the mRNA code for proline? So we know that tRNA has an anticodon which complementary base pairs with mRNA's codon. So therefore, we know that if GGU is the anticodon, then mRNA's codon must be CCA, as G complementary pairs with C and U complementary pairs with A. So therefore our answer is going to be B, CCA. The next question, which of the following base sequences is the DNA code for proline? Now I think this question is a bit ambiguous, to be honest, because when I think of DNA code, I think of the DNA coding strand. So therefore what I would do is I'd get CCA, which is obviously our mRNA code, work backwards to work out the template strand, and then work backwards again to work out the coding strand. However, this question requires that you basically provide the template strand code, so the complementary base pairings to CCA. So the answer is actually C, GGT, as C complementary base pairs of G and A complementary base pairs of T. But I don't particularly like that question, as I think it's a bit off-putting, but hopefully that won't come up again soon. So these are our answers. Question 8. Some antibiotics inhibit RNA synthesis and protein synthesis in cells. Actinomycin D, rifamycin and A-amanitin are antibiotics that work by binding to molecules in a cell. This inhibits protein synthesis. The scientists who developed these antibiotics had to find out which types of cell were affected and which molecule they were binding to. The table shows the types of cells that these antibiotics affect and the molecule that they bind to. So our first question is to explain the effect of actinomycin D on protein synthesis. So if we have a look at the table, we can see actinomycin D here, and it affects both prokaryotic and eukaryotic cells, and it binds to DNA. So if this molecule is binding to DNA, then we know that this is going to affect transcription. So the DNA strands won't be able to separate, which means that there's not going to be a template strand available. So this is going to obviously inhibit transcription, it's going to inhibit the mRNA synthesis, and therefore the DNA of both cell types are the same structure, so both cell types are going to be affected. And our next question, to explain the effect of rifamycin and A-amanitin on protein synthesis. So if we have a look at rifamycin here, so rifamycin affects prokaryotic cells only, and it binds to RNA polymerase, whereas A-amanitin affects eukaryotic cells and but also binds to RNA polymerase. So therefore, since they're binding to RNA polymerase, we know that RNA polymerase brings the RNA nucleotides, it lines them up against the template strand, and also it forms the final mRNA strand by joining the RNA nucleotides together. So for both of these, the effect is going to be transcription being inhibited or mRNA synthesis being inhibited. And because the RNA polymerase is inhibited, this means that it won't be able to bind to the DNA. So transcription is going to be inhibited because the RNA cannot bind to the DNA. And then you need to say about how each antibiotic is affecting different cell types because of the structure of RNA polymerases being different. So you can see our answers here. Question 9. The beta, delta and gamma polypeptide chains have similar amino acid sequences. The table shows the sequence of 9 amino acids in a part of each of these polypeptide chains. So then we've got a table with the type of polypeptide chain and then their corresponding sequence of amino acids. The beta, delta and gamma polypeptide chains are coded for by 3 different genes. Compare and contrast the sequence of bases in the DNA coding for each of these parts of the 3 polypeptide chains. So when it says compare and contrast, what it means is to give the similarities and the differences for the question. So if we start with the similarities, we can see that all of these have the same number of amino acids. So we've got 1, 2, 3, 4, 5, 6, 7, 8, 9 amino acids. So we know that there's going to be 9 triplet codons, or we could also say there's going to be 27 bases, because remember it's a sequence of 3 bases that codes from 1 amino acid. We can also see that there's a lot of similar amino acids in here. So we've got similar, we could say about PHE, so you just need to write the abbreviation. So you could say that all 3 will have the code for PHE or for amino acid 1, and we can also say that for amino acids 4 to 9 they are the same for all of the polypeptide chains, and we can also say that beta and gamma will both code for the same amino acid 2, as you can see here. And we can also say that delta and gamma will both code for the same amino acid 3, as we can see here. So those are our similarities. As for our differences, because they're being coded for by 3 different genes, as it says here. Oh, that's not the best colour. Let's get a different one. Let's get a light colour. Let's go for blue again. So we've got 3 different genes here. So that means that there could be a different code for the same amino acid. So we know that for a single amino acid there is more than one codon. The genetic code is degenerate. So the sequence of bases in the code for the same amino acid might be different. And then we can also say that because of the 3 different genes, the amino acids that are the same might have a different code. So for example, we could say that beta and delta will have different sequences for amino acids 2 and 3. Because these amino acids are obviously different, so they're going to have different sequences. And then we can also say how beta and gamma will have a different sequence for amino acids 3. And then we could also say how delta and gamma will have a different sequence for amino acid 2. So those are our similarities and differences, and you can see the answers here. Question 10. Scientists have discovered that the Zika virus genome is made of RNA. This RNA codes for capsid protein with 105 amino acids, pre-membrane protein with 187 amino acids, envelope protein with 505 amino acids, 7 non-structural proteins. State how many bases are needed to code for the capsid protein. So we know that, of course, it's a sequence of 3 bases that codes for 1 amino acid, known as the triplet code. So the capsid protein has 105 amino acids, so that means there's 3 times as many bases. So therefore, 105 times 3 is 315, so our answer is going to be 315. Part 2. Unlike human immunodeficiency virus, HIV, Zika is not a retrovirus. Describe what happens to the Zika virus once it is in the cell. So, the Zika virus, because it is RNA, we know that the RNA is in fact used in translation to make the viral protein. So, the RNA from the Zika virus itself will be used to make the mRNA strand, so therefore it will be used in translation, and this will make the viral protein. So therefore more RNA is going to be produced, and we can see our answer there. So thank you so much for watching. Please subscribe to my channel and make sure to give this video a big thumbs up if you have enjoyed it. Also head over to my Instagram and to my website, and you can access lots of exam questions and revision resources. Please let anyone you know who's studying GCSE or A Level Biology about my videos. I really want to help as many people as I can with their exams. So thank you again guys, and I will see you in my next video. Bye!